Today's webinar will cover a new workflow in our NGS applications, and it's the ability to combine analysis from different data sources into one place so that we can do things like compare gene expression and regulation together and visualize them together. So it's some really exciting new capability in the software. And so we'll spend the latter part of the webinar going through the software, and I'll show you how to set up the projects and do some of the analysis. I'd like to introduce, for those of you that are new to the company, we just have a few slides here where I can introduce you to DNASTAR. Our software is called Lasergene. The most recent version is 14.1. And we will have new software later this year. About twice a year we'll have different updates that bring new capability. And Lasergene software has wide-ranging functionality. Then I'll kind of give you an overview here. It can be things like molecular biology. So this is just some screenshots of our molecular biology suite that provide capability such as Sanger sequence analysis. I have some a screenshot here of some annotation results for plasmid annotation, multiple sequence alignments. So all the basic tools for doing analysis of sequences are part of our molecular biology suite. We also have the Genomics Suite, which is where we'll be looking today and focusing today. And our Genomics Suite offers both the assembly software, so the software that we'll use to take the raw input data, FASTQ files, align the data to a reference, and then provide a variety of different analysis options. And that's where we'll spend most of the time, is showing how to set up the projects and then the variety of different analysis tools. And we also have a structural biology suite, which allows us to do things like predict protein structures. So we may find proteins of interest through our genomics. We can focus then on the proteins that are affected. And these are some great new tools for looking at structures, visualizing them. And so, and of course, we have specific webinars that cover structural biology. So the vision for DNASTAR really is to provide a comprehensive set of tools in molecular biology, also have genomics, where we can do fast, accurate assemblies of transcriptomic and genomic sequence data, as well as protein tools. And really our goal is to unite all of those so that we can do all these analyses very easily and efficiently. And that's really what we're striving for. We have clients worldwide in academia and the government and pharmaceuticals. Our support network is wide-ranging. So we have distributors overseas. Our main office hours are Central Time, from 7 a.m. to 6 p.m. You can almost always reach a person here. So if you have technical support and have questions, you can reach someone here to help answer. We have chat as well, email. We also do webinars like this, where it's one-on-one with you or your group, and we can work through projects that you have and answer any questions that you have that way. So one of the focal points of our genomic software has been accuracy. Really in the last couple of years, we've been striving to be the most accurate assembler of NGS data that's available. And at this point, we're about 99.7% accurate on aligning data and finding known variations in that data. And we have extensive white papers on our website that show where the data sets are. You can reproduce this in our software. We have workflows built into our software for you to determine accuracy. You can use your own data sets as well. If you've got a set of SNPs in a data set, you can run it through our accuracy workflow, and it will produce the same numbers for false positive detection and as well as overall accuracy. So this is just a table here that shows how efficient we are compared to some of the more common genomic software pipelines. So, of course, BWA aligner with the GATK SNP caller is commonly used, and then as well as some commercial competitors, CLC and Genius. And this is on an exome data set. And what we can find pretty consistently is that we have a much lower false negative detection rate. And so that lower false negative detection rate gives us a better overall accuracy. So we can get a more accurate SNP call on these data sets. Now, the amount of time that we take as well, speed is another factor, not the top factor, but the elapsed time for exomes for us is anywhere from 45 minutes now to about an hour and a half. Our competitors are generally 3 to 6 times slower, just assembling and doing the SNP calls. And so we think we're in a very good position of having both the most accurate software and the fastest. And in terms of speed on other types of data sets, the assembly times range anywhere from a couple of minutes to several hours, depending on the size of the data. So again, very, very fast, very accurate, and the same algorithm that is used then for all our different NGS workflows. And today we're going to be looking at some workflows that are used for gene expression analysis as well as the regulation of those genes. And so we use that same underlying algorithm to do the alignment. We can also do SNP calls. So one of the combined analysis can be variant analysis combined with RNA-Seq or ChIP-Seq. Today we're going to focus on looking at gene expression, RPKM, calculated gene expression with peaks, and use some of the nice new visualization to show how we can very easily take very large data sets and combine them. So at this point, I'll jump out of our PowerPoint and we'll go into some software here. So the ability to combine projects is tremendously useful. Now I have some earlier webinars where we focus just on RNA-Seq, and we have some videos on ChIP-Seq. And it may be that you're doing RNA-Seq now, and down the road, weeks from now or months from now, you do some ChIP-Seq, and then you want to compare the two together. And so oftentimes with other softwares, you may have to just reassemble everything and somehow export tracks up to a genome browser. It's a very cumbersome and awkward kind of a workflow, trying to combine those analyses certainly with open source tools. With our software, we've streamlined things. And just to give you an idea, so if I did some RNA-Seq, and I did some earlier this month, and I can rerun that, but I'm just going to load this. So this is SeqMan Engine, and I'm going to go in here, and there's a script file that's left over from previous assemblies. I'm going to populate this wizard then with a script, and this was an RNA-Seq and reference-based, and I'm going to go through this fairly quickly. This is just an RNA-Seq setup, just to show you what I've done. And I use the human genome template package, so a certain build of the human genome. Right? And I loaded in some files, some FASTQ files, and some multiple sample RNA-Seq. And in this case, I'm going to remove the 12-hour time point. The demo that we have today is going to have a 0, time 0, and a time 24 hours. So I've got 2 time points, 4 different FASTQ files input, and 2 time points, time 0 and time 24 hours. And so I want to run a SNP caller and an RPKM normalization. Right? We'll name the project, make some wiggle files, and at this point, we're ready to assemble. Right? So I ran these assemblies before, and the output is on this computer, and it looks something like, and I'll just show you where these files went. Actually, I'm going to quit this. There we go. Oh, let's see here. Where is it hiding on me? Demo data, there we go. And so I'd run these RNA-Seqs, and so I get some wiggle files and the dot assemblies then for each time point. These contain the BAM files, all the alignment BAM files. So these were done back on June 6th, right? So they've been done already. And then I did a similar thing with the ChIP-Seq. So I'm going to open SeqMan Engine again, and I'm going to again load the script in so you can see what I'd done before. This gives me the ability to, so I load the script file in, and it just loads this wizard up to save us some time so you're not watching me load everything up. And so I can see, now this is a ChIP-Seq workflow, and same genome template. That's the most important part to know, is that when you're comparing RNA-Seqs and ChIP-Seqs or copy number variation or variants, you want to make sure that you're using the same genome template. If you use different genome templates, then you have to reassemble everything. But as long as you use the same genome, you can make these kind of comparisons. So this is a ChIP-Seq then. I have a control. I've got several different conditions. For the demo today, I just did a single a single ChIP-Seq with a single control. And we can mark the control. Again, this is just kind of rehashing. I can do a SNP caller here, normalize with RPM, and I'm going to use an E-range 3 peak detection for this data set. And there's max in E-range 2 and 3. And again, I won't go into details here, but the different peak detectors will have different parameters that we can adjust. And that's something that if you're doing ChIP-Seq, you'll want to read the help and figure out, what's the best peak detector for your particular type of ChIP-Seq experiment. Okay, so that's what I had done, is then we define the binding protein. It's this E2F1 binding protein, and I get a little Jasper logo here, and I can put a label on it. So again, typical kind of ChIP-Seq workflow. Name the project, and again, click Assemble, and what comes out is now I have ChIP-Seq assemblies right here. And I have the control.assembly, wiggle files, and I have dot assemblies for each experimental group that I ran. And these were also done in the same day, but they could have been done today or any other point in time. And now I want to come back and say, well, I want to compare those two together. I want to compare differential gene expression, for instance, changes in gene expression with binding events. I want to see, is my transcription factor, is it bound to the genes that are showing differential expression? So that's a question that I'd want to answer and do it as easily as possible. So if I go back here to SeqMan Engine and workflow, you can see I have choose assembly workflow, and then at the bottom is the combine and or reanalyze assemblies. And there's two options here. I can rerun or I can merge. In this case, I want to merge assemblies. And so I can merge them from different workflows. And what that means is it can be the same or different. So if it could be just RNA-Seq. I did 10 today. I did 10 assemblies a month ago. And I want to combine them together so that I can analyze them all together. So it saves me the step of having to realign everything. I can just use the existing assemblies and just rerun the analysis portion. And so, or I can just take existing and say, I just want to change how I did the RPKM or the peak detection on assemblies I've already run. I just want to change one parameter and see how that affects things. So this combine and reanalyze is very, very useful when you've run assemblies and you want to save some time and not have to reassemble them each time, but just redo the analyses or merge projects to do a combine. So it's tremendously useful. And so now I can add projects. So if I add folders that contain these projects, so let's go to the RNA-Seq. And I'm just going to load this folder, and it's going to look in the folder and see, oh, I've got 0, 12, and 24 hour RNA-Seq, and it automatically detects it. Now, just for today's sake, I'm going to remove one of them just to simplify it a little bit. And I'll go to ChIP-Seq as well. And so I had these ChIP-Seq folder here. And again, it'll look in there and see, oh, I've got a control and a couple of experiments, and I can remove some if I don't want to analyze all of them. So it's looking in these assemblies and recognizing what we've done. So I have RNA-Seq, ChIP-Seq. It knows there's a control for the ChIP-Seq. And now I get a page that allows me to set my signal processing. And so I can change what I had done before. So if I decided I don't want to do RPKM normalization for the gene expression, I could change it here. Or I could change the peak detection method. Or I can just leave it the same. I just want to combine them now into one. And so I've got several options here. So you could imagine if I did 100 RNA-Seq assemblies and I want to change one of the options. I don't want to reassemble. I just want to change an option. I can go through here and just change a single option for the analyses. So then we can just name this combined, and it's going to run this script again. So this wizard writes the script, and the script then tells the assembler how to do the assembly. Okay, so then it says start combining. So that took, it depends how big the data set is, but it can vary in time how long that's going to take. But it's certainly a lot faster than assembling all the data. So it'll run through, reanalyze everything, and then give us an output that is you're going to analyze in our RayStar software. And so I'll go right to that software. I've got that opened already, so I'll go to the first kind of page here. So RNA-STAR I have some floating windows here, and you can see the way it works, so there's tabs across the top that can set the views that we want to look at. So the experiment list then shows what experiments I have loaded into this project. So I have 3 different types of experiments. I have RNA-Seq, time 0 and time 24. I have a ChIP-Seq, and I have variant experiments for each one. So I also generated SNP tables for each one of these experiments. And so that, again, is just the experiment list. And I'm going to go now to the gene table. And we'll make this bigger. So this is kind of the starting point. And the gene table now is a combined gene table. Maybe I'll make this full size. And if I look in the status bar at the bottom left, I can see there are 27,787 genes in the gene table. So you start out with everything. And really the goal is to I want to find that interesting subset of genes that is both, there's a differential change in expression of the gene plus ChIP-Seq peaks nearby. And so somewhere in here, I should be able to find those. And I populated the gene table in a certain way. And you can see that I have a gene name. I have the RPKM values of times 0 and times 24. So those are the normalized expression values. I have a fold change column. Then I also have some functional information, functional description of that gene, and also a prediction on how, gene damage prediction. If this gene is affected by SNPs or some other kind of a mutation, what effect does it have on disease? So there's medium, high, low. We only see medium right now. And then on the last 2 columns, these are peaks. So these are the RPM values of the peaks called by E-range 3. And you can see the first thing that kind of pops out is, well, a lot of those are really low. There's no peaks by a lot of these genes because I haven't filtered anything yet. And so you really have to then apply some filters to find the subsets. And there's all different ways to do that in RNA-STAR. I'm going to show you just a kind of a simpler, straightforward way to apply some filters. And so I'm going to have a couple windows going here. We'll try to see if we can pull this off. So then I also have a scatter plot, and this is times 0 and times 24. And the scatter plot is nice, kind of like a sanity check. When I apply filters, I can look here and see, do I have the things that I want filtered, right? And what I really want to filter based on expression are these guys up here. So these are up expressed in 24 hours. Here's some that are down expressed, that go down from 0 to 24. And I don't want to look at genes down here because those are at really low levels. There might only be a couple of reads aligned to those genes. So the scatter plot allows me to kind of look at what my filters did. And you can catch mistakes very easily too. And so I've got some filters applied here. And the first one is a fold. So first of all, I should point out, I'm filtering all of them. So you always want to make sure that you're not just filtering a subset, so all the genes. I'm filtering genes, not peaks. And I'm matching all of the following. And I've added a filter, fold change. And so there's a bunch of different filters we could apply. So, I might use some of the annotations and filter against one of the annotation terms. In this case, I'm not. I'm just going fold change between the 0 and 24-hour time point, 2-fold higher in either, right? So I can do it, just up express genes or down express or both. So I just picked either. And then I added another filter, and this is expression level and signal. And I'll actually undo these so you can see. So if I just apply that filter, I'm looking in the status bar now. 6,829 genes are found. Right, so that's still a lot of genes. And if I click this little box, it'll show me, again, I call it my sanity check in my scatter plot. I can see that there's my fold change. You can see the fold change confidence bars, too, that are in the scatter plot. And it's like, well, that's not quite what I want. I don't want to analyze the low expressed genes here. All right, I just want to look at things that are expressed more at a higher level. So now I can add filters. And so gene expression level. And I'm just going to choose a signal here. It's essentially a lower threshold. And now let's just search right away and see. Should be some fewer now. So I look at that number. And as you make more complicated filters, it's easy to kind of make a logical mistake in the filter. So I like to look at it and say, yep, that makes sense. Let's add one more. And 24. And now we're at about 1,500. So we've got about 1,500 genes that show at least a twofold change that meet our minimum log threshold. So that's a pretty good number just based on possible interesting gene expression changes. So once you, you can keep filtering, but however you filter, at the end what you want to do is save this now as a set. So I'll just call this differentially expressed genes. I think I might have a set. So you can do this as many times as you want. And now it's just saving that set of genes to this set list. So at this point, I can apply other kinds of filters. I can say, well, now I've got my gene set. Now let's look at our peaks. So again, the combining is I've got both RNA-Seq and ChIP-Seq. And so when I ran my analysis, there were some thresholds that were set for peak detection. And so I look at this table, and there's not 27,000 peaks. There's a lot fewer here. There's 122 peaks that meet my threshold. And the threshold, you can see I've got a linear RPM value and a raw count. It's about 100 reads. And that's my bottom, my weakest peak has about 100 reads aligned to it. You might set a different threshold. I might just take everything that's here, but again, a sanity check now is, let's see what a peak looks like at this lower end of the range. So I'm going to right-click on it and send it to our new browser, which is GenVision Pro. And GenVision Pro then allows me to, it automatically navigates to that place from the table in RNA-STAR, and it shows me the data. And so I've got RNA-Seq data, so times 0, times 24. I've got my control for ChIP-Seq, and my peaks. And now I can zoom at different levels and see. And the first thing that kind of pops out is, oh, I don't really like the way that graph looks. So I can highlight an area and say, let's change the style so it's a little easier for me to, I don't want the grid lines in there. Maybe I don't even like the color that much. So I can change what this looks like. I can change the magnification scale as well. So you can see that we're zoomed in quite a bit. And as I zoom out, I can see even my range needs to be adjusted a little bit. So I can keep making adjustments and say, well, I want a little bit more of a Y scale. And I have my control, and I can see a peak there. It looks like it's at the interesting area possibly for 2 genes. I see the RNA-Seq peaks as well. And as I zoom out a little bit more, just kind of trying to go slowly here so I don't zoom way past, but you can see I can get more of the gene in, and I can see that ChIP-Seq peak kind of popping out, and I see these nice peaks for RNA-Seq. So that's at the low end of the scale. That looks sufficient to me. Now, I might go back and say, well, you know what? I want to have more peaks. I'm going to lower my filters and reanalyze my ChIP-Seq and try to get more peaks that are a little bit weaker to come into my analysis. So again, that's the advantage to just being able to reanalyze the existing assemblies, is I can recalculate the peaks that way. So let's go back now to RNA-STAR. So in this case, I'm just going to say, I want all these peaks. And I want a list now of all the genes that are somewhere near these peaks. And so there's a set of genes that are near those peaks of interest. And I can also remember them as a gene set. So now I have two sets, differentially expressed genes and genes with peaks. And now I can do things like make a Venn diagram and see, is there overlap? And so my Venn diagram now says, yeah, there are some genes that are differentially expressed and have a sizable peak somewhere near them. So I can select that intersection and say, show me the table of these genes. So here are those genes that meet both the criteria. And I'm going to do that again here, gene table. And I only want to show the selected genes in my table. So I've got 78 genes that are potentially interesting to me. Now this functional description, before we look at the peaks, I do want to comment on how I got these columns set up. I probably should have mentioned it earlier. But there is a column manager, and it allows me to create this table and display the current columns. I have the name, the RPCAM, a few annotation columns, and then I have the peak RPMs visible. But you can see the available gene info is all different tissue specific and from different databases. This information I brought in, I went to the DNASTAR website, and let's see, and on our website under support, there is a gene level annotation from DBNFSP, and I just downloaded that file and brought it in, file, import annotations. So I've just brought that file in, and that's what brought in all this additional functional type annotation information at the gene level. There are other ways, and if you have other databases, as long as the gene name matches up, you can bring it in through a custom import wizard. No problem. Okay, so I've got a set of genes now that are interesting both with respect to expression and peaks. Now I really want to look at them. I want to see, do they pass the eyeball test as well? So I have them all selected. And let's see here. And I want to send this to all of these to GenVision Pro. And so now it's going to, in GenVision Pro, there is a, let's see, maybe I can change this a little bit. There is a Places tab, and the Places tab has everything that was received sent from RNA-Star. So there's all the different genes and different chromosomes. And so I can navigate from gene to gene and chromosome to chromosome very easily now, just to those. I don't have to, use a browser to look up each spot individually. I can look at this first gene, and what I'm looking at now are, first of all, what do the scales look like? Do I need to adjust them? Right? What does my peak look like? Is it nice and distinct? And so I can very quickly evaluate then. See, I see a pretty decent peak right there on the 5' end of this gene. And I can change kind of the layout then. Let me just show you. So if I don't like, well, maybe I'll wait for one where we need to change it. So there's, and I might say, well, that's interesting to me. Maybe I want to go back and see, is that one, maybe I want to filter again and see, does that particular gene have involved in a pathway that I'm interested in? But I can say that's a favorite. I'm going to click this little star, and that'll add it to the favorites and say that's one I want to revisit. Now I can very quickly move through, and I can say, well, that one, there's a peak there, but it's a little bit noisy. Maybe that's not, I want real strong signal. So here's one. I can see a pretty nice ChIP-Seq peak right there. So I get a nice peak there. I can see expression. And I can just evaluate and go through. So here's one. Here's a histone. So it's got a broader peak. So I'm going to zoom out just a little bit. And I can see some of the peaks run off the top. So we can change that. And so you need, for these kind of displays, I need the ability to, change the y-axis. So maybe I need to go to 250. So I can really try to fine-tune that. That wasn't close to enough. So there's 1,000. Still not enough. So, well, it's 100. Let's go to 1,000 here. How about 10,000? So even at a much higher Y scale. So I can change the scales here to get this picture to look just how I want it. And I can go to the ChIP-Seq. I could do the same thing here. Make that scale 50. I can rename it. So there I have the, and I can call that rename that track, so it makes a little bit more sense. So I can get a really nice picture now. And now as I move, I can see there's a couple more histone type. Again, I can really nice controls for zooming in and doing analysis. There's another clean peak right there. All right, let's jump ahead to some genes that. And here you can see the peak there, so it's just sometimes just a matter of just navigating a little bit. And of course you see arcs there. Those are split reads that go reads between isoforms. So we have an earlier webinar that we did isoform analysis. I can move these tracks around, so I can click on them and put the ChIP-Seq right where I want it. Again, you'll want to change the scales here sometimes depending on the gene you're analyzing. So there I have ChIP-Seq on the top and RNA-Seq on the bottom. You see a nice peak right there, and I could keep adding these to favorites just like that. So it's really a very efficient way to start out with lots of data, RNA-Seq and ChIP-Seq, combine it, and start doing the analysis. So here's one with lots and lots of splicing and exons. Again, I can zoom right in on that. Now, if you get to a point where you want to take, images too, there's great options. So I'll change this one a little bit. So if you want to get that perfect picture for a poster or presentation, I have several different options here. So I can export the image, and this is the analysis, right? So I can do a PDF. I can do, save this as a PowerPoint. And let's throw that right on the desktop. All right, so I can save that range. There we go. So that's the image that's out. And of course, these are all objects too now. So it's so if I need to change text, I can change this again. Allows me to edit that image very easily. Every one of these little pieces is going to be editable. I can ungroup this, and if I need to fine tune this image, I can start ungrouping and make edits and make just the exact kind of image that I want. Okay, well, I know I threw a lot at you in the webinar. At this point, we're a little bit past our half hour that we had allotted, and I'd be certainly more than happy to stick around and answer any questions that folks have. So thank you again.
Thanks, Matt. Well, we had quite a few questions come in today, so because we're running a little bit late, we're just going to answer the first two questions, say. So how about starting with this one? How many experiments can be analyzed in RNA-STAR?
So RNA-STAR, so we look at something, just for a demo, we just had a couple of experiments, but RNA-STAR can analyze hundreds, so 100 to 300 RNA-Seq projects or exome projects or ChIP-Seq. So it can be scaled up quite large in terms of number of experiments that you bring in. So if you're, accumulating data, running, every week you run a few more RNA-Seqs, you can keep combining them, add them to that RNA-STAR project so you can do an analysis all in one place together.
Thank you. And also I wanted to let people know before I present the second question for today's webinar, we got in a lot of questions, so I'm going to ask Matt to answer some of those after the webinar via email. So if he doesn't get to your question, if your question is not the next one I'm going to ask, he will answer your question. It'll just be a little bit later on today. So thank you for posing those questions too. And the final question I'm going to ask Matt during the webinar today is, how long does it take to combine experiments?
So the combining, it really depends how many experiments, but pretty typically if you've got 10 experiments, 10 RNA-Seqs or 10 ChIP-Seqs, which would take roughly, say an hour to an hour and a half each if you reassembled them. The combining may take an hour to an hour and a half, somewhere in that range. So it's about a tenfold improvement in speed over reassembling everything, at least, maybe even a little bit more than that. So it's definitely a time saver. And again, if you're combining to large sets, if I've got 99 exomes and I'm adding the 100th one, then it's a lot faster. Adding that one to combine actually may only take an hour or so, whereas, of course, reassembling all those is going to take a much, much longer time. So it's a great time saver.